The CSEC by it of tomorrow.
Yeah, yeah.
Okay. Are we live? And I have with me here Miss Akili from the Student Up. No stranger to the YouTube lives. That's not, let me see the kind of settings that, oh, I could do this too. Oh.
Eh,
So, I'll know the drill. Exam tomorrow, so you're not going to waste no set of time talking and thing. Imagine having to do CXC. Imagine having to do CXC. Hey,
uh,
no, but biology is supposed to be, that's supposed to be normal thing, man, normal thing. But for those of y'all who don't find this normal thing, yes, I for a second I thought I might have had to bring the bio. I might have to teach you bio because 2 minutes is what ago is when Akili just touched down.
hey, listen. So I sent out the link for the live, right? And I'm like, any topics, guys? I plan to come here for a casual hour for a little small, probably do a little genetics question and move on. But when you see topics that are coming, so I'm like, all right, I need a PowerPoint. So I brew the coffee and I say, all right, let's go. My apologies. One time, this slideshow was made literally in the last hour. I pull from like crash courses I've done from since 2020 to now, you know. So like I just pull questions trying to cover the topics that people basically indicated. Um, so
All right, well, I leave you all in the capable hands of Miss Akili. Last time we were doing something in the live, Akili wasn't even watching the chat. So you see the chat now, Akili?
I am seeing the chat. I'm seeing the chat.
We have 630 people here. Let's press like on the stream, share it up. I've seen people
No,
shouting topics and thing, but we already picked the topics and
yeah.
set up the slideshow. So it's 2 minutes in. Akili, take it away. Y'all are waited it. I starting a chemistry stream 8 o'clock, but it looked like this by stream not going to finish. 8 o'clock. When the stream gonna finish? Like when?
I can't say, but what we can do is probably pause at 8 and then I'll probably come back later in the night or something.
It'll come back later. All right. So you can pause at 8, touch the chem, touch a little chem, and we'll come back with the bio. Don't worry yourself. Somebody says to do chem tomorrow, but I done link up with Miss already, and I done set the live stream. We can just do a little taste of chem. A little taste, and then we can come back with the bio. All right. So, Alia, let it flow.
All right, so guys, this is not a crash course. One, right? This is some set topics that people kind of indicated in the different group chats that I pulled from. And I'm going to try my best to go through as much as we can in the little time that we have available, right? I hope biology show you guys some love as well, too, right? So basically, from the syllabus, nobody asked for any questions from section A, right? When I ask, was it problems? So while I'm not going to do anything from section A since um put some respect on section A still, plenty marks going to come from section A, so make sure you review it, right? It's not going to be the majority of the people. No, it's not. The majority of the people will really come from section B. However, still throw your eye on section A, right? Don't just disregard it altogether. Okay? So moving on to section B, right? Moving moving on to section B. The first topic that was requested from section B was nutrition. Yes, there's a bio exam tomorrow. Now it's kind of late to be realizing that, right? So with the nutrition, at this point, I kind of left different annotations up so I could save my time from having to like write on the screen, right? So we just gonna keep this moving real quick. So the first thing in terms of nutrition, in terms of plants, you have to understand photosynthesis because plants are autotrophic organisms, which means that they're able to make their own food and they do that by the process of photosynthesis. So you should be able to define photosynthesis. And when you define photosynthesis, it is a process by which your plants use inorganic substances existing in nature, your carbon dioxide and your water, in order to make glucose and oxygen. While knowing this equation, you also need to know the chemical equation, right? So you can write this as a word equation, which is simply the words carbon dioxide plus water, giving you glucose and oxygen, right? You can write that out in words, not going to write it, basically. But then you also have to know the chemical equation, and the chemical equation must be balanced, which means that you see those sixes thrown in there. You have to make sure and include those in the equation. And balance just means that you have to have the same number of atoms on one side of the arrow as you have on the other side of the arrow. That's as simple as I can put it, right? So you must know the chemical equation. There are certain conditions that must be present for photosynthesis to occur. So one, you're going to definitely need light, right? You need light, you need chlorophyll. And there are experiments that you should be familiar with that proves that light and chlorophyll are necessary for photosynthesis, but you're going to have to find YouTube videos on that, right? We don't have the time to go through those, but familiarize yourself with those experiments, right? We're going to get to respiration just now. So we're going to get to respiration just now. So I'm not discussing that just yet. We're still in photosynthesis, right? I think that's it for what I wanted to discuss here. So then figure 2 shows a diagram of a cross-section of a leaf of a flowering plant. The leaf is the main photosynthetic organ of the plant. So the majority of photosynthesis is going to occur there, which means that that leaf is going to have a number of adaptations that make it well suited to its function, right? So you have the way they want you to label the structures A, B, C, and D. What I want you to pay attention here is that the lines point to specific cells, not to specific layers. So identify the cell and not the layer, right? So A, B, C, D. A is your upper epidermal cell, as I saw some of you talking about. B is going to be your palisade mesophyll cell. C, your vascular bundle, particularly the xylem. And D, your spongy mesophyll cell. So these are the various layers of your leaf. Your leaf is well suited to being the photosynthetic organ because one, it has a large surface area that is going to be able to intercept light. Light is important for photosynthesis. I'm talking too quickly. So I'm gonna slow down, right? So light is important for photosynthesis. The leaf is thin, right? Because you need carbon dioxide for photosynthesis, and that has to diffuse into the leaf through tiny holes at the bottom of the leaf called stomata. So your carbon dioxide has to diffuse in. Water is going to move up into the leaf through the xylem. So that's how you're getting your 2 raw products. You're going to get your carbon dioxide diffusing in, your water moving up via the transpiration stream to the leaf, and then so that's how you're getting your raw products, right? So the leaf is thin. It has a large lamina, which is the surface of the leaf, to be able to intercept as much light as possible. Over your upper epidermis, you're going to have a waxy cuticle, which helps to reduce the amount of water being lost by the plant, because the plant is losing water, it's going to put the plant under stress. So it also has adaptations in order to minimize water loss, right? In your various layers, your palisade mesophyll layer, this one is going to be the main layer where photosynthesis occurs, right? And the adaptations that those particular cells have for photosynthesis is that it has large prominent vacuoles in the middle of the cell that pushes all of your chloroplast to the periphery or to the border, allowing them to intercept as much light as possible.
Which one is where the guard cell? Somebody asked about the guard cell.
The guard cells control the opening and closing. So this is one guard cell here. The guard cells control the opening and closing of the stomata.
Got it.
Right? So yes, so the palisade mesophyll cell has a bunch of adaptations allowing it to maximize the interception of light. The more light you have available is the more the photosynthesis can occur. Your spongy mesophyll cell, you can see that all of those cells are much rounder in shape compared to the palisade mesophyll cell, and that allows for looser packing so that you can have these air spaces in this section to allow for your carbon dioxide to diffuse in. So sometimes they might ask you a question like, if all the air spaces was filled with water, what problems would the leaf face? And it's going to be getting the carbon dioxide to diffuse into the leaf, right? So there, that was that question there. How, if all the air spaces were filled with water, what you would do, right? So moving on to nutrition, looking at your various food tests, right? So you have your tomato fruit is placed in a juice extractor. To be quite honest, I could tell you about whatever type of fruit, right? Whatever type of fruit, whatever type of food, they want to test for your major food nutrients. So you need to know your basic food tests, right? So if you're testing, so no matter what type of food you're testing, they're going to look for reducing sugar, protein, lipid, and a carbohydrate, right? So if they're looking for the reducing sugar, you have to know and be able to describe that procedure, and you're going to use Benedict's reagent for your test reducing sugars. So you could test reducing sugars and for non-reducing sugars. So your reducing sugars are going to be all of your monosaccharides, and your non-reducing sugars mainly are going to be like your disaccharides. The reagent that you use is going to be Benedict solution, which is blue in color. For this test using Benedict solution, you must heat. Sometimes they will describe the experiment to you and they will leave out that heating step and ask you, well, will you see a color change or not? If it has no heat, if it isn't heated, the reaction does not occur and you do not see any color changes, right? So you have to be able to describe that. For protein, you're going to use, as I'm seeing in the chat, so what's it test for protein? You're going to use your Biuret reagent, which is also blue in color, but with your protein test, you don't require any heat, right? So you could just mix equal parts your Biuret reagent, equal parts your test solution, and observe for a color change. So both your Benedict's reagent and your Biuret reagent are blue, but the color changes are going to be different for both of them, right? So when you do your test reducing sugar, a negative test is that the Benedict solution remains blue. Positive test is any color change to either green, yellow, orange, or red. A lot of students are under this misconception that red is the only positive result for your Benedict's test, and that's not true. If it remains blue, it's negative. Only blue is negative. Green, yellow, orange, red, all positive. And the reason why we have this variation in the possibility of what positive could be is because this Benedict's reagent, this test is semi-quantitative, which means that the more reducing sugar present is the more red you're going to get, and the more red the solution will look. So breaking that down, Benedict solution is blue in color. If there's a small amount of reducing sugar present, there will be a little bit of red precipitate formed. A little bit of red in a blue solution is going to look green. Add a little bit more red, a little bit more reducing sugar, it's going to look yellow. Add a little bit more red, a little bit more reducing sugar, it's going to look orange. And if you add a lot of reducing sugar, then it's going to look red, right? So again, anything green, yellow, orange, red, all of those are positive, right? For protein, your Biuret reagent is also blue. So if it remains blue, that means no protein is present. People trying to talk to me on WhatsApp during the live, and right? Christiana, don't worry, girl. Lock in. You got this, right? With your Biuret reagent, your positive color change, if protein is present, what color you expect to see? For protein, you're expecting to see purple. So guys are trying to pack in as much in the time, but I am going to sit down because it makes no sense if you don't understand, right? So you're going to see purple. Now, purple is your positive result. However, remember, like if they show you pink is a very light shade of purple. So don't fall for that. Pink is still going to be a positive result. It just means that the amount of protein present is going to be a little bit, right? With fat, in this case, they told you that you have a watermark on the filter paper, which means that they did a grease spot test for fat. But you can also do an emulsion test as well. So with your grease spot test, that's when you're simply rubbing the piece of fat on a piece of filter paper, and you're going to look to see if the wet spot that is created, if that wet spot remains after 5 minutes. If it dries and the spot disappears, it means that the fat is absent. But if that translucent spot remains, it means that fat is present in the food, right? So that is how you test using the grease spot test. Your emulsion test, your fats are soluble in organic solvents. They are soluble in alcohol. So what happens is that you mix the food with alcohol to see if any fat in it is going to dissolve in that alcohol, and then you mix that alcohol with water. And if you see it looking cloudy, that's the emulsion forming. That's a positive result for lipids. If you mix the food with ethanol and then mix that ethanol with water and it still remains nice and clear, it means that that food did not have any lipid present, right? So know that even though this question didn't test the emulsion test, that is a second test for lipids that can come, right? And then your test for starch, which is the easiest and the simplest of them all, which is going to be using your iodine reagent. And iodine reagent is yellow-brown in color. And so if it remains yellow-brown, it means that starch is absent. However, in the presence of starch, it's going to make a blue-black color change, which means that starch is present. Okay? So that's your brief recap of your food tests. Moving along to the digestive system, right? You have to be able to identify the various organs in your digestive system. One is pointing to so it's coming. One is pointing to the liver. 2 is going to be pointing to the stomach. 3 is pointing to the leaf-shaped organ. That's going to be a pancreas. 4 is pointing to your large intestine. Your large intestine is also known as your colon. And 5 is pointing to your small intestine. So the process of putting food into the mouth, that is going to be called ingestion, right? That's where it starts. The food gets into your mouth. It's going to be chewed by your teeth, right? You have different types of teeth. Your incisors and canines to the front are going to be breaking off the food, bringing it into the mouth. Your premolars and your molars, they are going to be crushing and grinding the food, breaking it up from larger pieces of food into smaller pieces of food, which is going to be mechanical digestion, right? So you have mechanical digestion and you also have chemical digestion. Both of them begin in the mouth. Saliva produced by salivary glands are going to be mixed with the food as it's being chewed, right? Do we have to learn to draw this or just label? You should be able to sketch a layout as well as label, right? The, what was I talking about? So you have saliva being released into the food. Your saliva contains salivary amylase, which is going to be the first enzyme you meet, right? That is going to start your starch digestion in the mouth, right? The mouth has a pH of roughly 7 to 8, so it likes a neutral to slightly alkaline environment. That is going to be the optimum pH for the activity of your salivary amylase, right? Egestion is going to be different, and we're going to get to that to the end, right? So we have both mechanical and chemical digestion occurring in the mouth. What I want you to know and to stress is that mechanical digestion does not end in the mouth. It continues in the esophagus, in the stomach, and in the small intestine, because the entire alimentary canal is aligned with smooth muscle. So as the muscles are contracting and relaxing, they are also going to be helping to break up the food into smaller pieces. And why do you want to break up the food into smaller pieces? That is going to increase the surface area available to your enzymes, which carry out chemical digestion. Mastication is going to be that process of the food being ground and broken up into by your teeth, right? So mastication is basically chewing. So after you have chewed your food, mix it with saliva, you're going to form a bolus, which is a ball of food that you swallow. That's going to move down the esophagus into your stomach. Your stomach is a muscular bag that's going to be contracting and relaxing, churning and converting that food into chyme. your food could spend all the way up to between an hour to 6 hours in your stomach, right? It's going to then be slowly released into your small intestine. So now you have your liver and you have your pancreas, and both of them are part of your digestive system, but food actually doesn't physically flow through your liver or your pancreas. The reason why they're there is that they make substances, or juices as we like to call, that help in the breakdown, the chemical digestion of your food, right? So your liver is going to make bile and store it in your gallbladder. The pancreas is going to make pancreatic juice and release that into your small intestine. Somebody is asking what is peristalsis, and that is the action of your muscles, of your smooth muscles, that help to move the food along your alimentary canal. So that process of swallowing the bolus down your esophagus is contracting and relaxing the muscles of the esophagus to move that food down, right? So the bile stored in your gallbladder, the importance of bile is that it's going to emulsify fat, which means that it's going to take larger globules of fat and break it up into smaller globules of fat. Again, to increase surface area for your lipases. Lipases are enzymes that break down lipids. Once we get to, so your stomach produces gastric juice that contains enzymes that start your protein digestion. In your small intestine, you're going to have intestinal juice being produced by your intestine. You're going to have your pancreatic juice also mixing with the food there, as well as your bile. The small intestine is your main digestive organ where chemical digestion is going to be completed, right? What remains after that, your undigested material, is going to go into your large intestine or your colon, which is going to be important for absorbing things like water and any like vitamins and minerals that remain. You can have those being absorbed there. And as water is being removed from the undigested material, your waste is going to get progressively more and more solid. It's going to be stored in the rectum and then removed from the alimentary canal via egestion. Miss, what about the hydrochloric acid in your stomach? The hydrochloric acid in your stomach is important for killing any harmful substances in your food, as well as creating the optimum pH environment for the pepsin that has to start protein digestion in the stomach. So I have a next slide. State 2 reasons why enzymes are referred to as biological catalysts. In this question, you have to know that a catalyst is going to speed up a chemical reaction, but remain unchanged by that reaction. So that's what a catalyst is. It's going to be biological because it's naturally made in the body. So that's why it's biological catalyst. These are your enzymes. They are not changed in the chemical digestion process. However, they do speed it up for it to happen at a reasonable pace, right? The link at the bottom there, I already did a live with Mr. Springer where we were looking at focusing on enzymes. So if you need some more enzyme help, especially with regards to experiments, go check that one out after, right? So they want you to describe chemical digestion of your carbohydrates, fats, and proteins from the mouth until they are absorbed in the small intestines. And I'm going to, no apologies, there are a lot of words that are going to come at you here, right? So for your carbohydrates, in the mouth, it's going to start in the mouth with salivary amylase that's going to convert your starch into maltose. It's a stepwise breakdown. Starch does not go directly to glucose. It goes from starch to maltose. Maltose is a disaccharide, which is 2 glucose residues joined together, right? Then it's going to stop in the stomach because the hydrochloric acid in the stomach is going to make the pH be too low. Salivary amylase cannot work in the stomach. So starch digestion is going to start back in the small intestine, right? So stomach ulcers, your stomach contains this hydrochloric acid, your stomach is supposed to have a nice coating of mucus to protect the stomach from the strong acid it has in it. But sometimes the walls of the stomach can start to get damaged and you could develop stomach ulcers. Miss, move your camera, please.
Yeah, move it.
All right, help me out there. Right, cool. So in your small intestine, Your pancreatic amylase, which is going to be from your pancreatic juice from your pancreas, is going to continue any starch that was not digested in your mouth. So you're going to have more starch being converted into maltose, but the maltose isn't done yet. Then in your intestinal juice, you have other disaccharides that you consume. So the maltase that was produced from both your salivary and pancreatic amylase, they are going to be broken down into glucose. Glucose is a final product of digestion. Then you have sucrase, your normal table sugar that you use to sweeten your tea and your juice, etc. Sucrase is the enzyme that breaks down that sucrose into glucose and fructose, right? Glucose and fructose, final products of digestion. As well as you're going to have lactase, right? That is going to be the enzyme breaking down your milk sugar into galactose and glucose. Galactose and glucose are also final products of carbohydrate digestion, right? Then we have to talk about what happens to your proteins, right? Protein digestion does not start in the mouth, but it's going to start in the stomach with pepsin converting your proteins into peptides, which means that your proteins are going from large proteins into smaller proteins. SI is small intestine, right? Miss, when you finish your page, allow us to screenshot, please. Mr. Springer, there's any way for us to get this, I don't know, to make these slides available? No.
You could send it to Kavita and she'll put it on the Discord.
All right. That's
the
as
students have Discord.
yeah, so I can send a PDF of everything that you see on the screen, and you could pull it from Discord, as he says, right?
Yeah.
So yeah, so your pepsin is going to break your protein, your bigger proteins into smaller proteins. Then you have trypsin, also converting larger proteins into smaller proteins, but your smaller proteins are not fully broken down as yet, right? The 25 marks, Christian, that's a bit generous. I think it is. Let me see if I kept a copy of how much marks this question was for. I didn't. But it usually don't pass like a 6 to 8 marks, right? Miss, will the live be posted? Yes, it will, but I'm not too sure when, right? So we have your small proteins now. Then you're going to have your peptidases, which are going to break down the peptides into amino acids. So your final product of protein digestion is going to be your amino acids, right? Lipids, your lipids are broken down, which is probably going to be the simplest. It's going to be broken down by your lipases. Bile does not break down lipids. However, it takes the pieces of fat from larger pieces of fat to make it to smaller pieces of fat. Then, so we describe that as the emulsification of fat. Once that happens, then your lipases have a larger surface area to act on, and they are going to break up your lipids into glycerol and fatty acids, right? So that's where your glycerol and fatty acids will be your final product of lipid digestion. So you have to, where the highlighter, boy? Right. So you want to note these as your final products of your different macronutrients. Oops, no. Not maltose. Right, so that is digestion for those that asked. Moving on to the respiratory system. Some people ask for this. Figure 5 is a diagram of a human respiratory system. Name the label parts A, B, C, D, E, and F. So you should be able to label the various components of your respiratory system, right? So A is going to be pointing to your nasal cavity, right? Which you should, when you're breathing in, you should be taking air in through your nostril, not your mouth, right? So that's a question that they can ask. And the reason is because your nasal passages has a bunch of adaptations that help with the filtering of the air so they don't get particles, dust, and bacteria ending up all the way in the lungs, right? So that's something that you should note. B is going to be your pharynx, right? Which is going to be an area that is common to both with air coming from your nostrils as well as your mouth. Then you're going to go to C, which is going to be your trachea. And your trachea is held open by rings of cartilage, but not complete rings, C-shaped rings, because your trachea and your esophagus run back to back. So that the reason why you have the C-shaped cartilage is so that as whenever a bolus of food is passing, it's not going to be hitting the rings of cartilage all the way down, right? So that's just a question that could be an overlap between those two systems, right? So your trachea is your windpipe that is going to get air towards the lungs, and then that trachea divides into two bronchi. Bronchi is plural, bronchus is singular, right? So you're going to have your bronchus, and that's particularly pointing to your right bronchus, right? So in these cases, you might want to be specific when it's pointing to one side. So they're pointing to one, you say bronchus. If they had the lines pointing to the two of them, you would say bronchi. E is showing you your lung, particularly your right lung, and F is going to be your diaphragm, right? Is the larynx and the trachea the same thing? No. The larynx is going to be a voice box, which are some muscles that as you exhale, as air passes over it, it allows you to make sound, and that is what you use for talking, etc. Right? So no, that is not. Sorry, that should be, yes. That should be your left bronchus and left lung. My apologies. Let me fix that. Right? Thanks for that one. Right? So we also have to note about aerobic respiration. So respiration isn't simply just moving air into and out of the lungs. That's particularly breathing. So you should be able to distinguish between breathing and respiration. Respiration is the chemical process by which we take the energy that is stored in glucose and produce ATP, which are tiny packets of energy that we could use to pay for any energy-dependent reactions within the cell. All cells must make their own ATP. So that's why your cells must have your mitochondria, which will be the site of your chemical respiration. Right? So somebody is saying respiration is legit, photosynthesis inverted, and that could not be further from the truth. Come and join me for Cape Biology and you'll understand why, right? But in terms of looking at the chemical equation, which is a summary of the entire process, it can appear to be inverted, but the two processes are actually quite different. They involve different enzymes, different stages, etc. Right? Somebody asked a question and it disappeared, right? So you're going to chemicals. So this is the chemical equation. Yes, it still has to be balanced. So you're going to use one molecule of glucose plus oxygen to produce carbon dioxide and water. So these will be your products, but these are considered byproducts because the main point of this is to produce ATP to pay for your chemical reactions. The Discord link, Mr. Springer will, I don't know, help me direct you guys to where that will be, right? So differences between respiration and breathing, have a nice table here for you. So breathing is going to be is the process, or we also call it ventilation. That's just simply moving air into and out of the lungs, right? Respiration is taking the oxygen made available from breathing, getting it to the respiring cells so that the oxygen could be used to help liberate the energy from glucose to make your ATP that is going to be a tiny packet of, so that's going to be adenosine triphosphate. So, and you also should be familiar with your ATP ADP cycle, which I didn't have a question for, but it's basically whether ADP is when you have two phosphate groups attached, you use the energy from glucose to add a third phosphate group to give you ATP, right? I think on, that would be both on the Student Hub's TikTok and Instagram, I did a video on the ATP ADP cycle, right? Describe how oxygen in the atmosphere becomes available to human blood for delivery to all of your cells. Explain how this process differs from the way plants obtain oxygen for respiration. So very quickly, there's a common misconception that when you breathe in, you suck air in that's going to inflate your lungs like a balloon, right? That is not how air gets into your lungs, right? So it's kind of actually the reverse. You have your rib cage. Your rib cage has a bunch of muscles in it that you can contract to move your rib cage up and out or to drop it back down, right? Your lungs is vacuum sealed to your rib cage. It's not attached, but it's vacuum sealed to your rib cage. So just like I could attach this piece of paper to my mouth by sucking on the paper, I created a vacuum between my mouth and the paper, so the paper will stay attached to my mouth as long as that vacuum is there. And that is why your lungs remain attached to the, or adhered to the rib cage. So what you do to get air into your lungs is that you're going to contract some muscles, the intercostal muscles, that will move your rib cage up and out, right? Once that rib cage is up and out, your lungs are going to move with it. So your lungs expand. That drops the pressure inside of your lungs, and air is therefore going to be sucked in from the atmosphere to that low pressure environment, right? So I have that stepwise here for you. So your humans are going to inhale your atmospheric oxygen, which is going to be oxygen in the air. It's going to be inhaled through their nose or mouth when your diaphragm contracts. Your diaphragm is usually in a dome-shaped position. When it contracts, it flattens. So what that does is that it increases the space within your chest cavity, giving the lungs room to be able to expand, right? So by contracting your external intercostal muscles and your diaphragm, your rib cage moving up and out, your diaphragm flattening, you are increasing the space or the volume of the lungs inside of your chest cavity, right? That is going to drop the pressure and cause an inflow of air because the pressure inside of your lungs is going to be much lower than your atmospheric pressure. So the air is going to flow from high pressure to low pressure. So the air is sucked in, right? Once it's in the lungs now, it's going to be at the gaseous exchange surface. The alveoli within your lungs is the site of gaseous exchange. So the alveoli is where now your respiratory system is going to meet your circulatory system. You're going to have your, this question was 9 marks, right? So you're going to have your alveolus and your capillaries being over your alveolus, right? So that means the blood is going to be brought into very close proximity to the alveolus and the air inside of the alveolus, right? You're saying too much writing. Remember, you could get a copy of these slides from the Discord after the live is done, right? So I want you to focus on what I'm saying and understand what I'm saying, because just writing down these answers, I'm going to make the answers available to you, right? Right, guys, hold on. Hold on. So this is a topic that is common to both HSB and bio, right? So once you get the oxygen to the gaseous exchange surface, the oxygen in the air in the alveolus is going to be in much higher concentration than in the blood. So it's going to diffuse into the blood, right? So we're going to get the oxygen from the air into the blood. And once it's in the blood, that is part of the circulatory system that is going to get it to all of your cells. So the oxygenated blood now, where the oxygen is going to bind to the hemoglobin in your red blood cells in the blood, right? That is then going to go back to your heart via your pulmonary veins, and then your heart is going to push it out the aorta and all around the body, right? So then your capillaries are going to bring that oxygenated blood very close to all of your respiring cells to re-up them on oxygen and to take away carbon dioxide and other waste products.
I didn't get that. Could you try again?
See, reminding me business, guys. Yeah. So this is respiration. This is breathing, getting oxygen into the body, right? It's going to be fairly similar for getting carbon dioxide out. Once the carbon dioxide is brought in your deoxygenated blood, it's going to diffuse in the opposite direction. Right? In plants have a much larger surface area to volume ratio, which means that its surface area, its contact with the atmosphere, is a lot more compared to the volume of the organism itself. So it could rely simply on the diffusion of oxygen when necessary through the stomata, but it can also get that oxygen as a product from photosynthesis as well, right? So to be fair, this was one of the more challenging questions in terms of breathing. But the reason why I included it is so that you can have the steps that it would take to get the oxygen to the respiratory surface. So this is the respiratory surface zoomed in. This is going to be your alveolus here. And this is your capillary surrounding your alveolus, right? So you're going to have deoxygenated blood being brought here. Right? And at the end, you're going to have your oxygenated blood. Right? Your deoxygenated blood is going to be brought to the lungs via your pulmonary artery. Right? And it's going to have a high concentration of your carbon dioxide. Right? So once it gets there, it means that that carbon dioxide is going to be higher in the blood and lower in the alveolus. So A is showing you the diffusion of your carbon dioxide out of the blood and into the alveolus, right? Then that deoxygenated blood is also low in oxygen. So the concentration of oxygen in the alveolus is going to be high and in the blood is going to be low. So B is showing you oxygen diffusing down its concentration gradient into the blood, right? So this, not my mommy calling me in the middle of this live, right? So this is going to be high in oxygen and low in carbon dioxide, and that is going to go back to the heart via your pulmonary veins. Yeah? So Q here is showing you your red blood cell, right? P is your capillary, right? That will eventually join back up into the venules that will join up into your pulmonary veins. S is going to be the alveolus itself, right? And R is going to be the layer of moisture inside the alveolus, right? So that layer of moisture is important because the gases must dissolve before it diffuses across, right? Explain how human beings use anaerobic respiration during exercise. So the normal equation that we use for respiration is your C6H12O6 plus your oxygen to give you your 6 carbon dioxide plus 6 water plus your ATP, right? So usually oxygen is present, right? The red color is a bit hard to see in the green. All right. Well, I'll try to stick on the white because then it gets complicated, right? So instead, when oxygen is absent, aerobic respiration can't occur. So your body is going to resort to anaerobic respiration. And anaerobic respiration is a high interest, short-term loan, right? So it's going to help you out running a bind for a short amount of time. However, it's going to eventually work against you, and that debt has to be repaid pronto as soon as you stop exercising. So when you're exercising, your demand for ATP is going to increase because ATP is going to, I just block it with the text, right? ATP, can't see what the equation? Or the camera? E. Mr. Springer, you there? You have to pop me in another corner, please. Your whole mismove. I am not running the layouts, so Mr. Spring, I will have to move the camera. But what I could do is probably just write it to the bottom here. I think I'll have enough space, right? So C6H12O6 plus 6O2 to give you a 6CO2 plus 6H2O plus ATP, right? You can see to the bottom there. This is both HSB and biology, CSEC biology, but we're doing it here for the CSEC biology people that are writing tomorrow, right? So oxygen, so once you're exercising, your ATP demand goes up. So oxygen is going to be used at a faster rate. However, there's a limit to how quickly you could provide oxygen. And when that oxygen runs out, your body is going to opt to switch to anaerobic respiration. In anaerobic respiration, instead of breaking down the glucose into carbon dioxide and water, it's being broken down into lactic acid. Lactic acid is still a high energy molecule, so it means that you're not getting all of the energy that was in the glucose. So you're going to be producing a lot less amount of ATP compared to aerobic respiration, right? So this only produces 2 molecules of ATP for anaerobic respiration compared to aerobic respiration that you could have gotten anywhere between 36 to 38 molecules of ATP. So it's much, 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 much less efficient than aerobic respiration. So you should be able to make some general comparisons between aerobic and anaerobic respiration, right? Miss, do we need to know the amount of energy produced? No, so you don't have to know that specific detail. ATP and ADP, we talked about those a little while ago. But it's the energy currency of the cell. So the 2 molecules of ATP that you're getting allows the muscle to be able to contract for a little bit longer than it would have if we had just run out of oxygen and stopped, right? So it allows you to be able to contract for a little bit longer. Lactic acid isn't fully broken down, but lactic acid is also toxic to the body, which means that the more lactic acid you're converting glucose into lactic acid, the more you do that, it means that it's going to be building up within the muscle, and it even starts to diffuse into your blood as well. So this lactic acid starts to be circulating around the body, and it affects your brain and other organs, right? Not what you started.
We're taking a little pause in the next 10 minutes, swinging over and doing moles for chemistry, and we're coming back in an hour. That possible, Akili?
The hour, that will be 9 o'clock, a little tight. Like you could probably make it like 9:30 to be safe.
9:30. All right. Well, I certainly live 9:30. So we're coming back 9:30. So in 10 minutes, we're going to everybody. I know all you want to cram by right now, but Watch the chat. The chat like, no. So, Akili, and you have something to do right now or something like that?
Well, I mean, my babysitter is going to leave, so I have my children out there minding themselves.
Right, so we have to let Akili go and sort out her children. And then 9:30, she could come back. So while Akili gone, we now wasting time. We can hit them some chem. All you understand? All you understand? Chat, give me a chance. Give me a chance. All you don't understand too. so, 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 Akili, look my camera, doing all kind of stupidness. Akili, children to mind themselves outside it. So yeah, them had to figure it out, Akili.
Well, they had to tough it out. It looks so.
It will just be an hour and a half, and we coming back in, and we could go for long, right? Actually, after 9:30, we could go for long, maybe.
Yeah, after that, by the time we come back, we'll be focusing on section C. So I'll be doing the whole mitosis, meiosis. I'll be doing natural selection. I'll be doing some inheritance style questions. We'll focus on section C really coming when I come back.
All right, so we just go. So I don't know. It's just funny to me because the chat trying not to use obscenities now because the teacher. It's like, no, we do not want any chem right now. But Ali, we have to take a breather in an hour breather. But while we taking the breather, we're not going to waste time. We can hit a little moles one time. Even if you're taking it in full 100, you will be glad that we do that moles for chemistry on Monday when you come back on Saturday and revising chemistry, right? So she will come back fast, Miss will organize her kids, and she'll come back. So let me just get the rest of the next 9 minutes here, and then we can swing to chem.
All right, so we're continuing. So this lactic acid, it's not being fully broken down and it's toxic to the body. So your muscles going to start to cramp up. It's going to affect your brain, your vision is going to start to see black in the corners of your vision and stuff. And you're getting ready to pass out. You really are, right? That is only when you try to run your 5K for school and you never exercise or train for it, right? So, however, that lactic acid is going to force you to bring the kids to the library at a week, right? The lactic acid is eventually going to stop you from exercising, right? So that means that when you choose to stop or when your body stops you on the whole, you collapse, right? You're going to still continue to breathe heavy for a while after you stop exercising. And that is because you're going to still need the oxygen in order to break down the lactic acid so that you could fully break it down to get all of the ATP stored in it, right? While I'm on the break, I will send the PDF on the Discord so that you will have it when we pick back up at 9:30, right? Claire, you watching the kids? Right? I'll set up a little camera. So next I was going to start the circulatory system and we have 7 minutes, right? So I'll start and then we might likely have to stop. The break is going to be until 9:30. So figure 2 below is a diagram of a longitudinal section through the heart. Study the diagram and answer the questions that follow. Name the parts of the heart labeled 1 and 2. You're really making toast bread for my child for dinner. You have to be able to label the eye, yes. Right, so 1 is going to be your left atrium. And you're going to have 2 is going to be your left ventricle. Right? But while we're here, let's use the diagram to get a full recap, right? So we know that the 2 chambers at the top are going to be your atria. Atria is plural for. Atria is plural. The plural, right? Atrium is singular. Atria is going to be plural, right? So you have your left atrium, you have your right atrium. Your 2 lower chambers are going to be your ventricles, right? So this will be your left ventricle and this will be your right ventricle. So let's talk about how blood flows through the heart, and then we're going to talk about how the heart operates on a whole, right? So you're going to have blood coming to the heart via your vena cava, right? So that's going to be bringing deoxygenated blood to the heart, and it's going to bring it to your right atrium. Then it's going to flow through your tricuspid valve, and go into your right ventricle, right? Your right ventricle is going to contract and pump blood into your, this is going to be your pulmonary artery. And soon after leaving the heart, it splits in two to take one to your left lung and one to your right lung. So that's going to be to the lungs to pick up oxygen and to give off carbon dioxide. Vena cava is plural, vena cava is singular, but you have two of them. Right? So once it goes to the lungs, it picks up oxygen, it's reoxygenated. Then you're going to have your oxygenated blood coming from the lungs. Oops. From the lungs is going to come via your pulmonary veins. And it's going to come into the heart, into your left atrium. It's going to, do we have to say superior vena cava? So you have your superior vena cava and inferior vena cava. So I taught my students this so that they know it, but at this level, it's not really expected. But just know that you have more than one. So one bringing deoxygenated blood from the upper part of the body, and one bringing deoxygenated blood from the lower part of the body, right? So your left atrium is going to contract, and then blood is going to flow through your mitral or your bicuspid valve into your left ventricle. And then when your left ventricle contracts, it's going to push blood that then takes blood to the rest of the body, right? So that's how blood flows through the heart. What you have to realize is not that you have blood entering your right atrium, then that contracts into your right ventricle, so on and so forth. What happens is that both your atria contract at the same time to push blood into your right ventricle and your left ventricle at the same time, right? So both your atria is going to contract together. Then your both ventricles will contract, pushing blood into your pulmonary artery and the aorta at the same time. So your atria, when they contract, that's called atrial systole. And when your both ventricles contract together, that's called ventricular systole, right? So every time you have ventricular systole, that is what coincides with a heartbeat or your pulse, right? Well, particularly your pulse, right? So the atria contract, so the atria fill with blood from the lungs, from the rest of the body. Then both atria contract in atrial systole, pushing blood through your valves, here through your tricuspid valve, here through your mitral valve. Then once your ventricles filled with blood, they are going to contract after, pushing blood into your pulmonary arteries and your aorta simultaneously. So when you have that surge of high pressure blood being pushed into your aorta, that is what you feel as your pulse in different areas of your body where your arteries run close to the surface, right?
Arteries are way, veins bring blood to.
Yes.
Towards the heart.
Arteries are defined by blood vessels that take blood away from the heart. Veins are defined by bringing blood towards the heart. So we up on at 8 o'clock there, boys. So
Yeah, so you're gonna match the breaks here, yeah? Miss Christie's actually already live. So what y'all need to do is just search for the next live on my channel. Jump in for the break for Ken. And listen to me now. Have mercy. Jump in for the break for Ken. And we do Ken for 1.5. And then we coming back in with Bio and we'll finish off the night with Bio. Alright, so we're just gonna do some Chem molds. I know molds are a real tricky subject. I can't say relax with some molds in Chem because I know no relaxing. but we're gonna switch over, right? So I'll go and find the Chem live stream. I'm going to set up the live stream to start at 9:30. Miss Akili will be back at 9:30. Love and blessings, Alia.
see you in a bit.